Hi students, the last class we discussed something about the immunity. What are the types of immunity? The major types. One, the innate immunity or the natural immunity or non-specific immunity. Now we have to go through another type of immunity, what is called adaptive immunity or acquired immunity, also called by name specific immunity. Three names for that one. So anything which is acquired after birth, that is called acquired character. Likewise, the immunity that an individual acquires after birth is called adaptive immunity or acquired immunity. Acquired after birth, hence called acquired immunity. So it is a body's resistance to specific antigen or specific pathogen. So it is highly specific. For example, we'll talk about later under the characteristics what is specificity. So it is the body's resistance to specific antigen. Now this immunity has some unique characters or unique features. There are four important characters for this immunity. Number one, specificity. What's the meaning for that one? So it is the ability to distinguish differences among the various foreign molecules. For example, an antibody recognizes the tuberculosis bacteria. It also recognizes another one, say an example of leprosy bacteria. That is called specificity. Able to differentiate the different types of foreign agents, maybe a food item or a chemical or a medicine or the cells, anything else, the microorganisms, either the particulate antigens or the soluble antigen. They are reacting with only specific antigen, not all the antigen. That is called specificity. So it is the ability to distinguish differences among various foreign molecules. The foreign molecules I mentioned already, the different microorganisms or their products. The second one, diversity. The immune system can recognize a vast variety of foreign molecules. We have millions of using that for the microorganisms, either protozoans or bacteria or any other microorganism, their products, different types of antigens. So we have vast variety of foreign molecules all being recognized by what is called this immune system. What is what, whether it's a chemical or a microorganism or the products of microorganism, etc. And the third one, differentiation between self and non-self. So normally the immune system, when there is a foreign invader enters in the cell or enters in the body, they are being attacked by one's own immune system. This is a normal case. So for that it has to know whether the substance entered the body is its own substance or it is from outside, whether it's a foreign or a self molecules. So then only it can attack. So it is able to attack only the foreign substance, what is called the non-self molecules, molecules other than the body's host system, or immune system we can say. And see that one, if the system fails to distinct or fails to actually make distinction between self molecules and non-self molecules, then I mentioned already it resulted in the autoimmune disease. So for that it is one of the important properties of the immune system to differentiate the self molecules and the non-self molecules, the foreigner. Now the last one of the important mechanism operating the immune system is immunological memory. When any foreign agent enters the body, they are being recognized, they are being eliminated by the immune system. The immune system retains a memory of that first encounter for a prolonged time because we have different types of cells in the immune system. One type of cell in B lymphocyte, what is called the memory cells. Just like the brain, the memory cells retains a memory of the first encounter. When the same antigen, when the same microorganism enters the body again, now the memory cells recognize the antigen so that it eliminates the what is called the invader very quickly when compared to the first encounter. So that is called immunological memory. Now the immunization program or vaccination program is based on the memory of the immune system. This is one of the important questions also in NEET question paper. The vaccination or immunization for different what is called the diseases is based on the immunological memory. the memory of the immune system. For example, if you take the B lymphocytes, one type of cells, during the functional aspect, there are two types of cells are produced. One is called the effector cells, which produce the antibodies. Another one, what we call the long-lived memory cells. So the memory cells retain the memory of the first encounter. Is there is there any virus entered or any other bacteria entered? Which type of bacterium, which type of virus entered the body? That memory is there already in the memory cells. Nothing but the B lymphocytes. Also we have what's called T lymphocytes memory cells available. But the B lymphocyte memory cells are mostly responsible for actually retaining the memory and eliminating the invader by the production of what is called the antibodies through the plasma cells. We'll discuss later. So these are the four important properties of adaptive or acquired immunity. What are the components of acquired immunity or adaptive immunity? So it's being formed of two components. One, CMI. That is nothing but cell-mediated immunity. Cell-mediated immunity. Another one, antibody-mediated immunity or humoral immunity. What do you mean by cell-mediated immunity? Destruction of pathogens by cells. I am adding this word, by cells. By cells. Okay, without producing antibodies. The meaning for that one, the CMI is independent of antibodies. Without the help of antibodies, they will capture and then what we have, they destroy the microorganisms, anything else. They have some specificity, I'll talk about. So destruction of pathogens by cells without producing antibody. So it is antibody independent type of immunity. Antibody independent type of immunity. There is no involvement of antibodies. Only the cells are involved in capturing the microorganisms or anything else. Now the cells involved are macrophages, nothing but the WBC, the modified monocyte. The second one, T cells, thymocytes. The third one, NK cells, natural killer cells. These cells are large granular lymphocytes. Commonly called as LGL cells. So NK cells are large granular lymphocytes. Lymphocytes large in size with the granules. So they are called natural killer cells. So they kill actually, particularly the cancer cells or the infected cells. They never attack the viruses. They never attack actually, that is bacteria. They will normally do the action of killing the infected cells or the target cells by a variety of mechanisms. They are also involved in killing what is called the viruses and also the tumor cells. Okay, that's about the cell-mediated immunity, a specific type of immunity which involves mainly that is T cells along with other cells too. One of the important properties, working in an antibody-independent manner. Okay, the meaning there is no involvement of antibody. Another one, antibody-mediated immunity. It is also called humoral immunity because the antibodies are formed, dissolved in the plasma or serum. Some of the antibodies are also attached to the surface of the cells as receptors, but most of the antibodies are free antibodies circulating in what is called the serum or plasma. Hence called humoral immunity. Humor means the fluid formed, just actually scattered or dissolved in the plasma serum. Now, here it includes the mechanism which causes the destruction of pathogens and antigens with the help of antibodies, by the production of antibodies. Hence the name. So the antibodies are normally produced by B lymphocytes. The B lymphocytes are involved. Specifically we can see at the time of functioning, the B lymphocytes get modified into two types of cells. One, what is called the plasma cells. Another one, the memory cells. So this is nothing but the effector cell, plasma cells, nothing but the effector cells. The memory cells are long-lived cells, they can retain the memory of the encounter. So the plasma cells are nothing but they affect our B lymphocytes concerning the production of antibodies. Many times in the question paper, NEET question paper or general question paper, they asked about antibodies are produced by. We have the options B lymphocytes and plasma cells. The best option would be that is what is called the plasma cells. So the plasma cells can destroy what is called the type of just the plasma cells. They can destroy the pathogen by the production of antibody. So they are not working independently. They are working with the help of two major type of cells. Number one, what is called APC, antigen presenting cells. The antigen that enters in the body cannot be taken as such, cannot be destroyed as such, either with the antibodies or by means of some other phagocytic cells. They should be modified in such a way that they should be easily phagocytosed by the phagocytes. So, the antigen presenting cells, we have different types, dendritic cells, etc. They capture the antigen, then processing the antigen and presenting the antigen on the surface of them. Then only what will happen, the T cells are stimulated. So another type of cells involved in the case of antibody-mediated immunity, that is T helper cells, TH cells, otherwise called T helper cells. So once the antigens are processed and presented on the surface of antigen presenting cells, they react with T helper cells and further reactions are taking place in the immune system to eliminate the invader. Okay, now even in the case of babies, you know that one, the HIV virus attacks mainly the T helper cells, the switch, that's the main switch. Though it actually the place of production of antibodies B lymphocytes, the factory of the what is called the production of antibodies B lymphocyte. The HIV virus never attacks that one. It attacks only the workers, namely the T helper cells. Without T helper cells, the B lymphocytes or the plasma cells will not be stimulated to produce antibodies. That's why the HIV virus is always attacking mainly the T helper cells instead of attacking the place where the antibodies are produced. So anyway, the antibody-mediated immunity is brought about by the B lymphocytes, or we can say the plasma cells, and also we have antigen presenting cells. So antibodies are produced by the B lymphocytes, but the B lymphocytes is working through only this APC or T helper cells. That's about what we call the cell-mediated immunity, antibody-mediated immunity. Will have more mechanisms. We'll discuss later about all these things. Okay, let's pass on to the next one. Now let us see what are the types of what is called the acquired immunity or adaptive immunity. There are two major types. One is active, another one passive. What do you mean by active immunity? So it is an immunological response developed by an organism by the production of antibodies in their body. So antibody production is taking place within the body of affected organism, not gained from outside. Antibodies are produced inside the body of the affected organism. So the immune system of the host body produces antibodies. So how is it acquired? It is acquired through the use of person's immune response. Person's immune response, not from outside. Once antigen enters in the body, the meaning for that one, it stimulates what we call the antibodies production, and that is fighting against antigen. That's why the name antigen is derived from, you know that one, antigen, it is nothing but working against the antibody. So it is normally called antigen, that is referring to antibody generated. Antibody generated, a little later about that one. Why is it given the name antigen? Because it generates antibody. So antibody generator. I'll write out later the meaning. So it uses the person's own immune system, that is a host immune system, to produce antibodies. The result is eliminating the invader, at the same time the development of immunological memory. That is nothing but the development of memory cells. Both B lymphocyte memory cells, T lymphocyte memory cells are produced to retain the memory of that encounter. It results from, how is it developed? It results either from recovery from a disease, or we can say simply an infection. Suppose a person is infected with the tuberculosis or any other disease. So he can get what is called the active immunity. For example, the smallpox. There's no smallpox now. Similarly, a person developed chickenpox. After recovery, he developed immunity power against what is called chickenpox, and that is called an infection. Or this is a natural method. Recovery from the disease, development of what is called the immunity, that is nothing but a natural method, natural later about. Or by means of artificial method, we have a number of immunization program. By means of vaccination or immunization, we can just impart immunity. So this immunity is developed either naturally or artificially. By natural method, after recovery from the disease. By artificial method, by means of immunization for different diseases. For example, you have vaccine for the 3 diseases, like that. Polio vaccine, like that. So that's about what we call active immunity. So the duration is now, we will compare also separately what are the differences between these two. This is only the definition. Now passive immunity, it does not require the body to produce antibodies to antigen. No production of antibodies by the body to antigen. But how can we just actually oppose the antigen? The antibodies are introduced into the organism from outside. Such antibodies which are prepared in other animals' body and then introduced into the body of the host to develop immunity. Such antibodies are called preformed antibodies. Preformed antibodies. So in passive immunity, the antibodies are developed in other organisms' body in the blood, and then the antibodies are collected and then they are introduced in the body of an organism to impart immunity, and that is called preformed antibodies. For example, ATS. This is what is called anti-tetanus serum. Anti-tetanus serum, which is prepared by the introduction of what is called tetanus bacterium into the body of horse, so that the horse blood or plasma develops antibodies. The antibodies are collected from the blood or plasma of the horse and then being introduced into the body of a human to impart immunity. That's why I said here in passive immunity, but it's not long lasting. It's only of short duration. So the antibodies are introduced from outside into the organism. Nothing but the antibodies prepared in other animals' body are being used to impart immunity. Such antibodies prepared in other animals' body are called preformed antibodies. Now how is it occurred? So there is no activation of the immune system. So it is not necessary for the immune system to be activated to produce antibodies. So it is occurred without the activation of person's immune response. So that there is no development of memory. It doesn't lead to the development of memory. Here participation of the host immune system which leads to the development of memory, but here there is no participation of the host immune system. There is no response, there is no activation, and so also there is no development of memory. That's all about what we have the different types of immunity. Again, in the case of active and passive, we have further classification. I want to tell you 1 or 2 points. Now, active immunity is further classified into natural active acquired immunity, then artificial active acquired immunity. What is natural active acquired immunity? It is developed after recovery from the disease or an infection. So for example, a person developed immunity against chickenpox after recovery from chickenpox. So he developed immunity, natural. Then artificial one. This is by means of immunization. So artificial active acquired immunity is obtained by immunization. We have different vaccines available. So by means of vaccination, we can prevent, that is, the development of disease and also impart immunity. Likewise, in the case of passive immunity, again, natural passive acquired immunity, then artificial passive acquired immunity. So the natural acquired and natural what is called acquired passive immunity. Now it is the mother who transfer the antibodies. The antibodies are already prepared in the mother's body. Through placenta, the antibodies are transferred to the fetus. For example, IgG antibodies. And likewise, all through the breast milk, the colostrum. The colostrum contains what is called IgA antibodies. So either through the placenta or through the breast milk from mother to fetus naturally. That is called natural passive acquired immunity. Another one, artificial passive acquired immunity, I mentioned already. The antibodies are prepared in other organisms. Say an example for tetanus. We have prepared what is called a serum, anti-tetanus serum, anti-serum. That is being developed in the heart's body, then taken out. That is being used to impart immunity in the case of human beings. Such antibodies which are prepared in other animals' body, they are called preformed antibodies. So artificial method involves the introduction of preformed antibodies into the recipient's body to impart immunity. The example is anti-tetanus serum. So here antibodies are prepared in others' body, others' animal's body, and transferred to the recipient system. And here it is from mother to fetus. That is called natural method. And likewise here also we have active immunity. There are two types. Okay. Now we have to go. We already studied. We have to make the differences between the artificial and then what is called, I made a mistake. We have to make the differences between the active immunity and passive immunity. What are the differences? Nearly 6 differences are listed in the book. I want to discuss here. Now let's compare the active and passive immunity. Number 1. The active immunity occurs actively by the host immune system. There is a participation of host immune system. But in the case of passive immunity, it occurs passively and no host immune system participation or activation. It's not being activated. There's no need. The second one, it's being developed through contact with an antigen or pathogen. But here I mentioned already, the preformed antibodies are transferred from outside into the organism. Then it's long-lasting, durable, long-time effect and effective in protection. But this one transient. The word transient refers to temporary. For example, cholera vaccine. It's an example for passive immunity. Typhoid vaccination, example for passive immunity. So it is temporary and less effective. Now there is a memory system. Immunological memory is present in the case of active immunity. What I discussed earlier, there is no immunological memory because there is no participation of the host or recipient's immune system. So we are giving booster doses. So the booster effect on subsequent dose is possible. First dose, second dose is improving, increasing the intensity of what is called immune system, the immune power against the disease. But subsequent dose is less effective. Whether you are giving it or not, its effect is very less. So for example, we are giving the polio drops, booster doses. That is effective. It adds, booster means is that it adds more and more immunity power. But here it's not effective. The last one, it requires some short duration for its development. It's not immediately active. The period, what is called the lag period. So it is effective only after a short period of time, not immediately active. But whereas a passive immunity is effective immediately but is being lost within a short period of time. So it is very effective within a short period of time. At the same time, it is not short-lived. It is being lost quickly. For example, cholera vaccine, typhoid vaccine, etc. All being lost within a short period of time. We cannot give the booster doses also. That's about the active and passive immunity, the differences and the comparison. So we have to just go for the immune responses. There are 2 types of immune responses. One is called the primary immune response, another one is secondary immune response. Now what is primary immune response, what is secondary immune response, and also the differences between these 2. Let's make. What are the responses shown by the immune system as a result of entry of pathogen or any other microorganisms? So there are 2 types of what is called immune responses. One primary response, another one secondary response. What is primary response? The one which is developed first. When the immune system is exposed to the antigen or immunogen for the first time, that results in primary response. See, it occurs when the pathogens come in contact with the immune system for the first time. So what will happen during that what is called primary response? So during this time, the immune system has to learn and recognize about the antigen, what type of antigen that enters, learning and recognition. Then the second step, production of antibodies. The third step, production of what is called, that is memory lymphocytes, to retain the memory of that encounter. So when compared to the second response, it is very short time, that is normally the duration is very short, that means it's short lived and also very slow, occurring very just gradually. Now the antibodies involved in the primary response or IgM time. IgM time. Whereas here the antibodies involved in this response or IgG type. We'll discuss later. Now what is secondary response? When the immune system is facing the same antigen again, now the secondary response develops. Because already the memory cells develop, they retain the memory of the first encounter on the contact of the antigen or exposure of the antigen to the immune system. Now the memory cells already there. So it occurs when the immune system faces the same antigen again. For example, any virus, Nipah virus. Once again the Nipah virus enters in the body. What will happen? The secondary response, it's the highest immune response when compared to the primary response. What is happening during this time? The immunological memory has been established strongly because to develop the antibodies, to just find out or recognize the antigen. Then the second one immediately start producing the antibodies. Within hours after the recognition of antigen, a group of army plasma cells are produced, a group of new army cells. Army cells means they are nothing but actually the plasma cells which are fighting against the antigen. They are concerned with the production of antibodies. So formation of new army plasma cells to fight against antigen within hours, within short period of time. Then also within 2 to just actually 3 days. The concentration of the antibodies in the plasma or in the blood increases steeply and is reaching the peak much more than that of what is called the primary response. During primary response also the antibodies are produced, but the concentration of antibodies here is reaching a peak when compared to what is called just the primary response. Then what will happen? That is, as a result of antibody increase, it will fight against antigens very quickly within a short period of time. Now the secondary response is also called as a booster response or an amnestic response. An amnestic response. So I mentioned about IgM, IgG, these are all the types of antibodies or immunoglobulin fibers we can study later about. Now antibodies are produced only by the vertebrates, not by invertebrates or not by any other animal groups. So antibody production is concerned with only the vertebrates. Okay. Now let's have the comparison between the primary and secondary responses. Okay, I'll make it now. Let's have a comparison between the primary response and secondary response. The primary response occurs when there is a primary contact with the antigen. That is the first contact. Now the secondary response occurs as a result of second or subsequent what is called contact with the antigen. That's the same antigen. Contact with same antigen, not different antigen. Now antibody level increases. It takes more time, 7 to 10 days. But for antibody level increases within 3 to 5 days in the case of secondary response. The third one, the time taken to establish memory is longer. So a prolonged period is required to establish memory. But this one establishes memory within a short period of time. Then antibody level declines, decreasing very rapidly. But the antibody level there remains same for a longer period. Then it occurs in which organ, in which lymphoid organ. Now the primary response occurs in lymphoid organs like spleen and lymph nodes. Whereas this one, it first occurs, the secondary response first occurs in just actually the bone marrow, the primary what is called actually lymphoid organ or central lymphoid organ, followed by the secondary lymphoid organs like spleen and lymph nodes. So the bone marrow is the primary lymphoid organ, that occurs first, and followed by the secondary lymphoid organs, spleen and lymph nodes. We'll talk about later about this one. Now what type of antibodies involved in primary response? This is a neat question. So the primary response involves antibody IgM, mu. Then here the antibody involved in second response is IgG, the most common type of antibody, the only antibody that can pass across placenta. That is called IgG antibody. We'll talk about under immunoglobulin, the molecular structure, the antibody. So these are all the comparisons between primary and secondary response. Now in the next class we have to talk about the lymphoid organs. What are the types of lymphoid organs? Primary, secondary, etc. Okay, thank you.